And we saved Ed Silverman, who is not Canadian, unfortunately, but is a respiratory physician and professor at Harvard University and the Peter Brigham Hospital. He's at the Channing Laboratory, where he directs the Division for Network Medicine, and he's been the leader in the study of genetics of COPD for many years now. And he's a genetic epidemiologist, and he's the co-investigator founder of the COPD gene study. And so we're looking forward to hearing about the genes which drive the mRNA, which drive the protein.
Well, thank you, Peter. It really is a great pleasure to be back in Vancouver and to be here to celebrate this wonderful event in Jim's honor. He's just been such an inspirational leader to me and so many other investigators in pulmonary research that we're just so grateful for all that he's done. All right, so I am going to talk about the genetic epidemiology approach to COPD, and not with that. Uh-oh, that's a bad sign. So these are my conflicts. So why would we think that a disease like COPD that has such a strong environmental influence would have a genetic basis as well? Well, first of all, the development of COPD in smokers is quite variable. If you look at lung function values in families from the general population, they tend to cluster. If you look at COPD, it also tends to cluster in families. Now, environmental factors like smoking cluster in families too. So just because there's familial clustering, it doesn't prove that genes are involved, but it's a necessary prerequisite. There have been large twin studies like this study a couple of years ago in Denmark and Sweden that have shown that potentially as much as 60% of the variation in COPD could be due to genes. And of course, a small percentage of COPD patients inherit severe alpha-1 antitrypsin deficiency, which proves that genes can influence COPD. All right, let's try to switch over. So this is the overview for how we go about trying to find genetic determinants for a complex disease, because COPD is almost certainly influenced by multiple genetic as well as by environmental factors. We typically start by showing that there's this familial clustering or aggregation, and historically we've used 4 approaches. We used to spend a lot of time collecting extended pedigrees to do linkage studies and to try to do positional cloning. That worked extremely well for monogenic disorders like cystic fibrosis, but was a disaster for complex disorders like COPD. We also used to think it would be smart enough to guess what the candidate genes were going to be in the disease of interest. In general, we weren't smart enough to do that. So most of the studies have been recently genome-wide association studies, or with the falling costs of DNA sequencing, the ability to actually look at rare variants as well. No matter which approach we're using, we're trying to localize the general area where the susceptibility gene is located. But then we still have more work to do. We have to identify what the functional variants are and then figure out how they impact the disease. If we can do that, we could develop new diagnostic tests, figure out how much of the disease is explained by particular variants, and have new pathways for treatment. There are about 10 million common genetic variants in the human genome. But fortunately, we don't have to study all 10 million because nearby variants are correlated. And if we pick a carefully selected set of about 500,000 genetic variants, single nucleotide polymorphisms, or SNPs, that will allow us to capture most of the common genetic variation in Caucasian or Asian populations. Association studies determine if a particular genetic variant, either an allele or a genotype of that variant, occurs more often in subjects with a disease or other phenotype of interest. And genome-wide association studies, or GWAS, are really just made possible by the technological advances that allowed us to test hundreds of thousands of genetic variants at reasonable cost. So testing several hundred thousand genetic variants allows us to capture most of the common genetic variation for a disease of interest like COPD. Why would we care? What is the potential utility of knowing about genetic determinants? Well, it has the potential to help us learn about new biological pathways in disease pathogenesis. I think this is the part that I find to be most compelling for complex disease genetics, because we can really think of genetic variants as nature's experiments, nature's perturbations of the underlying biological networks that cause disease. This potentially could lead to new targets for drug development. We've been especially interested in how genetic factors could allow us to reclassify complex diseases, similar to the approach that Avi mentioned for using transcriptomic information to try to find molecular subclasses. We'd like to really classify the disease based on the cause of the disease and the pathophysiology of the disease, rather than an end-stage physiologic measurement like spirometry. And we think there's the potential for pharmacogenetics. We'd like to be able to find patients that are likely to have an excellent treatment response and treat them, and to avoid treating individuals who are at high risk for adverse events. Ooh. Okay. Well, we've spent a lot of time building study populations for the last 20 years for COPD genetic studies. I started about 20 years ago, as Peter mentioned, with that Boston early onset COPD study, selecting people with severe COPD at an early age and then getting as many of their relatives as we could for linkage studies and now for DNA sequencing studies. Also, we're fortunate to get DNA samples from about 400 people in the National Emphysema Treatment Trial. Controls were not enrolled in that study, but we did get controls from the normative aging study. We were pleased to collaborate with investigators in Norway on the Genkol study of about 800 cases and 800 controls. We worked with Dr. Perret and others in Vancouver on the International COPD Genetics Network, a nuclear family study with more than 3,000 people. Also, we're pleased to collaborate with Dr. Coxon and others on the ECLIPSE study, a study primarily of COPD cases, but with a modest number of control subjects as well. And then, as Peter mentioned, we've most recently been focusing on the COPD gene study, which has more than 10,000 smokers. Boy, okay, I think we're just going to stay here. Using these study populations, we're able to look at both candidate genes and genome-wide association studies. One of the most natural candidate genes, of course, is alpha-1 antitrypsin deficiency. We know that severe deficiency in individuals who typically inherit 2 of the Z alleles are at markedly increased risk for COPD. And a natural question was, are people that inherit 1 of those Z alleles at increased risk? So there have been more than 100 studies that have looked at this question, and the evidence has really been equivocal. Craig Hirsch and our group almost 10 years ago did a meta-analysis looking at studies that met various quality control standards. And he showed that studies that were case control studies, comparing COPD cases to controls, tended to find more MZ individuals in the COPD cases. But if people looked at population-based studies and compared FEV1 levels in relationship to genotype, there were very similar FEV1 levels in MZ and MM individuals. So it was hard to reconcile these 2 kinds of data. Inga Cecilia Sorheim came to work with us a couple years ago and showed that there was a reduced FEV1 to FVC ratio in MZ smokers, both from that Genkol study in Norway and from the International COPD Genetics Network families. And interestingly, there seemed to be increased emphysema in the MZ subjects with the lowest smoking exposures. In the Sepaldia study, Toon and colleagues showed no overall effect of the MZ genotype on FEV1 decline, but they did show some evidence for increased FEF2575 decline in MZ people that continued to smoke. Now, in the COPD gene study, we were able to look at a large number of individuals. One of the problems with the Z variant is it's rare, only a allele frequency of about 1%, and typically that's been hard to study in association studies. But with more than 10,000 people, we've got 256 MZ individuals in the non-Hispanic white portion of COPD gene and 22 African Americans who are MZs as well. And we looked at the presence or absence of COPD, emphysema, and gas trapping based on CT scans, and FEV1 and FEV1 over FVC. In each case, the MZ individuals in the non-Hispanic white group had increased risk for COPD or COPD-related characteristics. And in the African Americans, despite the tiny sample size, we still saw significant effects on lung function. So I think there's growing evidence that the MZ type really is a significant risk factor for COPD and one that we need to take seriously. Sorry about this. Well, the GWAS era for COPD started in 2009 with companion papers that came out in PLOS Genetics. Wilk and colleagues did a GWAS in that Genkol's Norway study in about 800 cases and 800 controls, took the top 100 SNPs and tried to genotype them in the International COPD Genetics Network families, and then did further validation in our National Emphysema Treatment Trial cases and normative aging study controls. They found a region on chromosome 15 in a cluster of genes that include several components of the nicotinic acetylcholine receptor and an iron-binding gene called IREB2 that met genome-wide significance. And when we're testing all these SNPs, we need to have a very stringent level of statistical significance to know that we've really found something. So typically we require a p-value less than 5 times 10 to the minus 8th to declare something statistically significant. It's a stringent bar, but this region met it. There was a nearly genome-wide significant region on chromosome 4 near the HHIP gene that I'll discuss in some detail. That was actually the region that did meet genome-wide significance in the companion paper by Wilk and colleagues that started by looking at FEV1 over FVC in the Framingham Heart Study, and then replicated findings in the Family Heart Study. All right, so this is the HHIP region. This is sort of our luck in COPD. We wait 20 years to find a COPD susceptibility gene, and it ends up being in an intergenic region. Right? So here's the strongest evidence for association. It's located between these genes, but there's clearly something in this area that influences COPD susceptibility. And we'll, in a few minutes, talk about how we're trying to move forward to understand what that is. It's a different problem in the chromosome 15 region. Here, the problem isn't being an intergenic region, it's being in the middle of many genes. So here we've got several components of the nicotinic acetylcholine receptor that bind nicotine and are clearly related to nicotine addiction and lung cancer and peripheral vascular disease. But there's also this iron binding gene, which we've, through other evidence, come to believe may be involved in COPD pathogenesis as well. Well, we know that larger sample sizes are important for finding new genetic determinants in COPD, and Michael Cho led a collaborative GWAS that included nearly 3,000 COPD cases and about 1,400 smoking controls. That led to the third GWAS region in a gene called FAM13A, a gene of unknown function. Not surprisingly, the other previous loci were supported as well, since similar populations were used. We focused on this FAM13A locus because it was a new association, and we're able to replicate it in several other populations. So again, meeting that very stringent statistical significance threshold overall. So this then was the third genome-wide significant locus for COPD. The strongest evidence is in an intron of that gene of unknown function called FAM13A. FAM13A just means family of unknown function, family 13 of unknown function. But we don't know for sure that it's actually even this gene that's being influenced. It could be a long-range regulation of another gene. What's been reassuring about these first three COPD GWAS regions is that in contrast to the candidate gene era, where one person would publish an association and someone else wouldn't be able to replicate it, there's been fairly consistent replication of these three regions. So for HHIP, it's typically been the same SNP, the same direction of effect for replication. FAM13A, either the same SNP or a nearby SNP. The story on chromosome 15 is more complicated, likely because there probably are multiple genetic loci in that region, but consistent replication has been shown. Our most recent efforts in these COPD GWAS has been with the COPD gene study, looking at the full 10,000 subjects in the COPD gene population. This is actually the subset of non-Hispanic white individuals that Michael Cho analyzed. And when we did the GWAS, we were a little disappointed that we didn't have any novel regions that met genome-wide significance. We had strong associations to the three previously reported GWAS regions, and that was confirmatory, but nothing novel. But this is why it helps to have friends. We were able to combine the results with the ECLIPSE study, the Norway study, and the NET-NAS study. And when we put all this evidence together, we now have overwhelming significance for those three previous GWAS loci and a new locus in chromosome 14 within a gene called RIN3. We decided that it might be worthwhile to look at severe COPD, recognizing that even though our sample size would be smaller, people with severe disease in GOLD 3 and 4 might be enriched for genetic factors. So we decided to compare those severe COPD cases to our control subjects, and indeed the strength of association for the three previous GWAS loci increased with stronger odds ratios and lower p-values. And we now had two novel GWAS regions, one in a cluster of matrix metalloproteases, including MMP12, and another in TGF-beta 2 that reached genome-wide significance when we replicated it in the COPD genetics network. So this is the kind of results that we get from these GWAS studies. This is the MMP cluster. You can see there's a bunch of genes in this area, including MMP3 and MMP12. So somewhere in this region is something that influences either one of these genes or another gene nearby, or potentially not so nearby. Now, what do we know about these three new COPD GWAS regions? Well, we know a lot about MMP3 and MMP12, that region, especially MMP12. This goes back to work that Steve Shapiro and others did in the 1990s, showing that an MMP12 knockout mouse was resistant to development of emphysema in a chronic smoke exposure model. Matt Honeyhockey showed that an MMP12 genetic variant was associated with lung function and COPD in a range of different studies. And then investigators here at UBC, by Drs. Ishii, Perret, and Sanford, recently had a very nice paper showing that MMP12 mRNA levels in alveolar macrophages were associated with both FEV1 over FVC and DLCO. TGF-beta 2 is interesting. It's in some ways been the neglected younger sibling of TGF-beta1. It has not received nearly the attention that TGF-beta1 has, but this region nearby has been associated with FEV1 over FVC in the general population lung function GWAS studies of charge and sporomata, and it is secreted by airway epithelial cells in response to injury or inflammatory cytokines. RIN3 is a gene of unknown role in lung disease. We did find that there were lower mRNA levels of RIN3 in COPD lungs compared to normal lungs. And of interest, it's located about 1.7 million bases upstream from the alpha-1 antitrypsin gene. So whether this is some kind of a long-range regulation or another protease inhibitor in that area remains to be determined. Well, in parallel to these COPD case control GWAS studies have been the large GWASs of lung function in general population samples in the Charge and Sporomata Consortium. In this study led by Solar Ortigas and Nature Genetics a couple years ago, they included more than 48,000 individuals with GWAS and replication in another 46,000. Very simple phenotypes, prebronchodilator, FEV1, and FEV1 over FVC. They found 16 new genomic regions. Interestingly, one of them was TGF-beta2, and confirmed 10 previously demonstrated signals, including HHIP and FAM13A. So there are an increasing number of these lung function genetic determinants that seem to also be COPD genetic determinants. Now, how many of those are going to end up being COPD genes remains to be seen. There are similar associations in the ever and never smokers, so I suspect that many of these genes influence lung growth and development rather than COPD pathogenesis, but there may be important areas of overlap in recapitulation of developmental pathways. We've started to move the other direction, to go from the genetic determinants we're finding in COPD to see what role they have in people with more normal lung function. So in COPD gene, we looked at the GOLD 0 to 1 individuals, so individuals with normal spirometry or mild airflow obstruction, and just counted up the number of COPD risk alleles at 4 different COPD loci, the FAM13A, HHIP, CHRNA3 locus, and this chromosome 19 locus. And the larger number of alleles that you've got, the lower your FEV1, or the higher your emphysema. It's not quite as good as it looks, because there are very few people that have 0 or 8 alleles, but it is statistically significant in terms of the relationship of lung function in people without classic COPD. So where are we in terms of the current top genes for COPD susceptibility? I think there are some definite regions that at least I would argue strongly for. Some of these come from Mendelian syndromes like alpha-1 antitrypsin deficiency and cutis laxa. Elastin and fibrillin 5 mutations can cause cutis laxa, which often includes emphysema. From the COPD GWAS, there are these 3 regions that have been replicated in multiple studies at strong levels of significance, including HHIP, FAM13A, and this chromosome 15 cluster, and now 3 new regions in MMP12, TGF-beta2, and RIN3. And other genes that are sort of not quite to that level of certainty, but likely will, with additional studies, reach that degree of confidence, either from lung function GWASs or other COPD GWASs. So I think we're making progress in identifying COPD susceptibility genes. But although these initial association studies are useful and helpful, it's really just the first step. It's what I like to call the discovery process in genetic epidemiology, which needs to be followed by localization and functional validation. Localization can involve studies of fine mapping by testing more SNPs or DNA sequencing or testing in more populations, looking for long-range genetic interactions, because many of these GWAS hits will be regulatory genetic variants as related to gene expression, as Avi has described, and also trying to find regions that contain functional activity. We also need to do functional validation with both cell-based models and animal models, and we've typically been focusing on the murine chronic smoke exposure model despite all of its vagaries. So let's walk through the process for HHIP, this region on chromosome 4, where we've got this block of genetic variants that track together. Starts about 51,000 bases upstream from the HHIP gene. The gene on the other side is called glycophorin A, and as I mentioned, it's in an intergenic region. Well, we thought that it was probably, we then needed to say, well, what's going on here? So we've got this intergenic region, where are the functional variants? Well, maybe they're actually within the HHIP gene. Maybe there's an amino acid changing SNP in the gene itself or something in the promoter, and we're seeing a long-range correlation with the genetic variants upstream. Maybe there's a novel gene in that region that just hasn't been described yet. Maybe there's a novel functional element, a microRNA or some other genomic regulator that hasn't been described. Maybe there's long-range regulation of HHIP. That's our leading hypothesis and one I'll try to convince you of. Or maybe there's long-range regulation of another gene. We thought it was probably going to be an influence on HHIP regulation because we saw differential gene expression. If we looked at COPD lung tissue compared to control lung tissue, we saw lower levels of mRNA and protein of HHIP and COPD lungs. That gene on the other side of that LD block, glycophorin A, wasn't even expressed in lung tissue. We went on to do fine mapping studies. This was work led by Michael Cho, and what he did is he did genome sequencing of this 228,000 base pair region, including that upstream GWAS region, the intervening space, and then all the introns and exons of the HHIP gene. He found 493 SNPs, but importantly, there were no common amino acid changing SNPs that could explain this association signal. So it didn't seem like it was going to be some sort of long-range correlation with an amino acid changing SNP within the gene. We thought it was going to be more likely a long-range genetic regulator, and Xiaobo Zhu and our group used an approach called chromosome conformation capture, or 3C, to see if there was evidence that this upstream GWAS region was folding over and regulating the HHIP gene itself. So we used the HHIP promoter as the anchor, and then put some intervening primers here with denser coverage of the LD block of association, and looked to see if there was evidence for long-range regulation of any of these regions. This work was done in two different cell types, a BEAST2B, a lung epithelial cell line, and MRC5, a lung fibroblast cell line. You can see there's a strong interaction of the HHIP promoter with itself. So the y-axis is interaction frequency. The x-axis is the distance along this chromosome 4 region. So positive control, strong interaction of the HHIP promoter. Then an absence of interaction. Then a second peak of interaction right within that GWAS region that then disappears. So this 7 kb primer within that GWAS region appears to be folding over and interacting with the HHIP promoter. Further work narrowed that 7,000 base pair region down to a 500 base pair region that contains enhancer activity. So when we put that 500 base pair region in front of the HHIP promoter, the expression of that HHIP promoter was increased. There were two SNPs that we had found from our sequencing efforts in that 500 base pair region, and we put those two genetic variants together in front of the HHIP promoter. There was reduced expression, again, not dramatically reduced, but modestly and statistically significantly reduced expression. And one of these genetic variants appeared to change the recognition sequence for a transcription factor, the SP1, SP3 family of transcription factors. So we went on, and by we, that is clearly the royal we, because I don't do this kind of work. But Becky Barron and Xiaobo Zhu in our group used electrophoretic mobility shift assays to show that there was increased specific binding to the C allele, that's the COPD associated variant compared to the wild type T allele. That could be competed away by an identical competitor, but not by a non-identical competitor or a 6 base pair mutation of that region. And when we incubated with an antibody against SP3, we saw a supershift suggesting that there was specific binding to the COPD-related genetic variant. So we think we've found at least one of the functional variants in this region that influences HHIP and may be involved in why there's lower HHIP expression. But we don't really know what HHIP has to do with COPD pathogenesis. So we decided to go ahead and use RNA interference, knocking down the expression of HHIP, and then try to see what biological effects that had. Our hypothesis was that since HHIP is part of the sonic hedgehog pathway, a critical pathway for lung growth and development, that we would silence HHIP, and we would primarily see effects on other hedgehog pathway genes. So we used RNA interference in BEAST2B cells, ran a microarray experiment, did RT-PCR validation in BEAST2B cells, and then RT-PCR assessment in primary lung epithelial cells, and then went on to look in lung tissue samples. So this summarizes the gene expression microarray results. We knocked down HHIP just as we had planned, analyzed it a couple of different ways, found 300 genes that were differentially expressed, and none of them were in the hedgehog pathway. So contrary to our hypothesis, this was not primarily a hedgehog effect. We looked carefully at the members of the hedgehog pathway with real-time PCR and saw some subtle effects on GLI1 and GLI2, a couple of the downstream hedgehog pathway members, but at least in BEAST2B epithelial cells, this was not primarily a hedgehog pathway effect. What we saw instead was that there were a range of extracellular matrix genes that had differential expression after successfully knocking down HHIP. These included an elastase inhibitor, elafin, PI3, Syndican1, Wnt5b, so a range of things related to extracellular matrix and a little beyond that. And also we saw differential expression of cell survival and proliferation genes with a range of genes either mostly down with decreased expression after knocking down HHIP, but BTG2 going increased with expression. And so some of the downregulated genes, including BRC5, BIRC5, and TGM2, these are important cell survival and proliferation genes, and most of them were validated in primary lung epithelial cells. So we took these differentially expressed extracellular matrix genes and cell survival and proliferation genes, and we decided to make a network. We worked with John Quackenbush and used a variety of different kinds of evidence from the literature and from known functional relationships, and the purple circles here correspond to differentially expressed genes. With the more purple it is, the greater the differential expression. And what we found was there were a lot more connections between those extracellular matrix genes and those cell proliferation genes than we would have expected by chance. Now, what's also useful is that we're now starting to see some of the genes that we think were more likely to be involved in COPD. So we've got MMP9, part of the matrix metalloprotease family, EGR1, a key transcription factor that's been related to COPD, integrin alpha V is over here somewhere, that causes emphysema in a knockout mouse model. So as opposed to the sonic hedgehog pathway, which would be harder to relate to COPD pathogenesis, it seems like this gene may have additional functions related to extracellular matrix and cell survival and proliferation that could allow us to fit it into more traditionally known pathways for COPD pathogenesis, or even better yet, extend those traditional pathways in more useful ways. We've also been working on the HHIP knockout mouse. It turns out that the homozygous knockout is embryonic lethal. So we've been working with the heterozygote knockout mouse in work led by Xiaobo Zhu and Carolyn Owen. As you know, the chronic smoke exposure mouse chronic smoke exposure model after 6 months causes a modest increase in mean linear intercept in the wild-type mouse. That effect is exaggerated in the HHIP heterozygote with an increased amount of emphysema. So despite the relatively modest reduction in gene expression in these heterozygote knockout mouse, which is probably fairly similar to the effects of the SNPs in a human being, we do see some effects in this mouse model. In the last couple minutes, I just want to shift gears and talk about a theme that I'm pleased to hear that several other investigators have mentioned, the idea of COPD heterogeneity. And we can think about this heterogeneity in a range of different ways, as you've heard about, but we like to think about it as both COPD subtypes, trying to understand distinct groups of COPD subjects who may have different amounts of emphysema and airway disease, the underlying processes, or different degrees of COPD-related disease manifestations, so differential exacerbation frequency, cachexia, functional impairment, and comorbidity. In the COPD gene study, we have parallel efforts going on to try to understand COPD heterogeneity using clinical subtypes with imaging-based expert classification and epidemiologically driven analysis of COPD subtypes. And also, what I'll tell you about today, our work in machine learning approaches, which has been primarily based on trying to cluster COPD into meaningful subgroups of patients, but also to try to define disease axis using approaches like principal components and non-negative matrix factorization, because maybe we're not really going to find discrete subsets of COPD. Maybe we're just going to find continuous axes, and different people are going to be at different locations on those continuous axes. Michael Cho and Peter Castaldi have led this cluster analysis work. We've got more than 10,000 people in COPD gene. This analysis focused on the 8,128 with complete data for all the clustering variables that were used. They split their data set into a training and a test data set, which I think was important. It hasn't been done in many other clustering studies. And based on a range of analyses that I don't have time to go into, picked 4 key variables: FEV1, percent emphysema using a minus 950 Hounsfield unit threshold, emphysema distribution as the ratio of upper lobe to lower lobe emphysema, and airway wall area of segmental airways. Using those 4 variables, they found that there were 4 clusters. One was a set of resistant smokers who had normal spirometry and no emphysema. Second was people that had relatively normal lung function, but an upper lobe predominant emphysema distribution. The third group had even lower lung function and the thickest airways and no emphysema, so this would be an airway predominant group. And the fourth group had the most severe reductions in FEV1 and the most emphysema, so a severe destructive group. We were able to use additional clinical and genetic information to try to validate these clusters. So compared to that resistant smoker group, there were increased amounts of exacerbations, increased shortness of breath, differences in Bode score, and increased hospitalization rates in all three of the other clusters at strong levels of significance. But I was more interested in the genetic associations where HHIP, for example, and the chromosome 15 SNP had differential associations to these clusters, with the cluster 2, that mild upper low predominant group, having the strongest associations to HHIP, and the chromosome 15 region having the strongest associations to the severe destructive emphysema group. And these odds ratios are stronger than we saw in the whole population, suggesting that if we have well-defined subsets of COPD, we may see stronger genetic effects. Where I think we need to go is to have parallel efforts that will use both biological samples and improve phenotyping, where the phenotyping will involve imaging, physiology, clinical assessments, with approaches like machine learning and clinical approaches to come up with disease subtypes, to have biological samples that will enable us to do whole genome sequencing in a range of omics analyses, from transcriptomics to proteomics to metabolomics, to use that information in an integrated network analysis approach to try to find the disease determinants. And then in an iterative way, frankly, we haven't worked out how to do completely yet, come up with new approaches to disease classification. So I think that genetics has the potential to help us understand COPD. It has the potential to identify subgroups of COPD patients with different etiologies and potentially with different pathophysiology. It has the potential to help us identify new mechanisms of disease and to identify new targets for treatment intervention, but we still have a ways to go to make that a reality. I'd just like to acknowledge my many collaborators on this work. I'd be happy to answer any questions.
For a question or one? Yes, Jim, you had a question?
Yeah. Really interesting talk.
Is your mic on?
Yep.
Good.
Now, what I wondered, I know in the COPD gene that you have the ability to define the COPD into the emphysema phenotypes. Now, is there, when you take a different emphysema like paraseptal and compare that to one of your other emphysema phenotypes, is there anything like in the various levels? Is there anything that sort of comes out so that these might have different representations in the sites you found?
Great question, and something we're very interested in doing. One of the limitations we've had so far is that in the first phase of the COPD gene study, we don't have visual assessments of the emphysema patterns yet. We're in the process of doing that, but it's going to take a while. So this is where we've been fortunate to use approaches like George's group have developed with the local histogram approach to try to assess what the pattern of emphysema is related to, and the severity of it and the amount of it in normal, mild central lobular, moderate central lobular, severe central lobular, panlobular, and paraseptal, or plural based. And we're just starting to do those genetic analyses. I think that so far I can say that I think they're going to give us somewhat different signals than the analysis of emphysema overall, but it's a little early to see exactly how that's going to play out. But it's what we need to do.